Today we will talk about the potential of non-invasive photobiomodulation therapy for neurodegenerative diseases via neuromuscle interaction. First, what are neurodegenerative diseases or NDDs? Simply put, they are caused by the progressive damage and death of our nerve cells. This leads to a gradual loss of functions like memory or movement. Common examples include Alzheimer's, Parkinson's, and ALS. The key challenge is that disease don't have a single cause. As you can see, neurodegenerative involves a complex mix of problems such as protein aggregation, mitochondrial dysfunction, and neuroinflammation. The traditional approach to treating these diseases faces two major problems. First, the wolf problem. It's extremely difficult for drugs to get past the blood-brain barrier, or BBB. This means many potential treatments can't even reach their target inside the brain. Second, the focus problem. For decades, our strategy has been almost exclusively brain-centric. However, entities are also heavily influenced by the crosstalk between the brain and peripheral organs. So this brings us to our core concept of neuromuscle axis. This communication happens at the neuromuscular junction, or NMJ. In diseases like ALS, the problems often starts right here at the NMJ before the main nerve cells in the brain is damaged. This means the muscle is an active player in entities. How can you strengthen this axis? The most powerful and well-studied answer is exercise. When we exercise, our muscles release beneficial molecules called myokines like the BDNF irisin. These myokines deliver to brain, promoting brain's plasticity and help grow new blood vessels. So exercise can affect to protect the brain. But this raises a critical question. What if a patient is too old or hard to exercise? And that leads directly to our new topic, PBM. Photobiomodulation, PBM, applies specific wavelengths of the light to stimulate cellular function non-invasively. PBM targets the mitochondria, enhancing ATP production and modulating the reactive oxygen species, ROS, similar to the initial cellular responses observed during exercise. This activation can lead to the upregulation of key transcription factors like PGC1 alpha, which is also activated by exercise and is involved in myokine production. And this leads us directly to the central goal of our project. We will track how PBM changes key functions in muscle, such as mitochondrial health and myokine release. Then, determine if these muscle-driven effects can ultimately provide neuroprotection and slow down the disease.
Our overall research approach focuses on evaluating how peripheral PBM influences mitochondrial function and brain health. First, we will examine mitochondrial changes, specifically mitochondrial membrane potential and RS levels, which reflect energy status and redox signaling. We will also evaluate cognitive functions through behavior tests like the open pill test and radial arm maze, which assess memory, anxiety, and motor activity. Finally, we will assess pathological changes, including myokine and neurotrophic factor levels, as well as markers of neurodegeneration, such as neural loss, inflammation, and blood-brain barrier disruption. Zooming in on mitochondrial function, we will quantitatively assess how PBM affects three key aspects. First, mitochondrial membrane potential, MMP, will be measured using JC1 or TMRM to track changes in energy capacity. Second, we will measure RS levels using DCFDA or MitoSox, since transient RS production can trigger protective signaling. Third, we will evaluate mitochondrial biogenesis and mitophagy by analyzing markers like PGC1 alpha, TFAM, and Parkin to determine if PBM promotes mitochondrial turnover and renewal. This analysis will allow us to determine whether PBM creates a healthier mitochondrial environment that supports brain resilience. To determine the neuroprotective effects of peripheral PBM, we will combine both behavior and pathological assessments in a neurodegenerative mouse model. First, we will evaluate cognitive and behavioral improvements using tests such as the open-field test, which assesses spontaneous locomotion and anxiety-like behavior, and the radial arm maze, which measures spatial learning and working memory performance. These tests will allow us to detect subtle improvements in the brain function following PBM treatment. Second, we will examine neuropathological changes in the brain. Specifically, we will quantify the levels of alpha-synuclein, beta-amyloid aggregates, and plaque accumulation, all of which are key pathological hallmarks in neurodegenerative disease. In addition, we will assess neuron loss and structural degeneration, as well as markers of blood-brain barrier leakage and neuroinflammation using techniques such as neurohistochemistry, immunohistochemistry, and ELISA. Together, this behavioral and molecular analysis will help us determine whether peripheral PBM can induce meaningful neuroprotection and improve both function and pathological in the diseased brain. We recognize several potential pitfalls in our approach. First, peripheral PBM may have limited systemic impact and it may not offer sufficient direct neuroprotection as a standalone treatment. Also, the molecular mediators like specific myokines will remain unclear and difficult to pinpoint. To address these challenges, we propose three strategies: using PBM as an adjunctive therapy, adopting a physiological resilience model, and applying untargeted omics analysis to identify key mediators. These alternatives will help expand both the application and understanding of PBM. PBM is non-invasive, accessible, and has a low risk of side effects, making it a strong therapeutic candidate. Clinically, it represents a novel approach for treating neurodegenerative disease and may improve quality of life with better tolerability. Importantly, PBS stands apart from conventional neuropharmacology and holds market potential as a safe, innovative therapy. Thank you for listening.